collect blood. So there is some kind of blood sample taken, then blood sample is analyzed for various analytes, right? That's what it comes to your mind. But of course, clinical diagnosis actually involves many things, but significantly blood analysis. Okay, so you may actually see that you in fact have several things in blood. For example, you have blood gases, electrolyte, metabolites, proteins, DNA, pathogens, and then properties of the blood itself. Okay, so I hope you have actually come across such kind of classification of what is the constituents present in blood and how we want to quantify it and why is it important to quantify. Okay, so now we'll slowly go into the topic. Well, as far as diagnosis is concerned, as far as utilization is concerned, diagnosis, you will do it for disease diagnosis, or you may actually want to screen the population. For example, nowadays, COVID screening, serological survey, ICMR is conducting serological survey. So that is actually for screening the population. How much of the population really is getting affected by this COVID, and then monitoring. So you want to, for example, you take another case, you want to monitor disease progress, okay, or disease treatment. So how treatment is actually working. So you want to do that, okay, monitoring. So as far as result is concerned, it can be yes or no detection, or many times it has to be quantification. Okay, so these are some small introductory concepts that you are very much aware of. I don't need to tell you, but we do have many other signals originating from the body. For example, electrical signals, especially people from computer science, they may be interested in actually getting these signals out and analyzing these signals. For example, you have a pattern for each and every signal. For ECG, for example, has a pattern. EEG, electroencephalograph, may not really have a pattern, periodic pattern, but it may actually give you a lot of information which doctors really go through for several hours together to analyze. So one can actually simplify doctor's job by actually doing some signal processing of EEG data. Okay, whereas EMG, electromyogram, that is the electrical signal originating from the heart, from the muscle. ECG from heart, EMG for any muscle. So there is really no pattern to EMG signal, and you simply see the intensity and other frequency. That's it. And endoscope, you actually typically do an endoscope. Endoscopy, there's something called as endoscopy, so that you will do mainly to find out if there is any abnormality with respect to that organ. And then some imaging techniques, they're all there. So yeah, but when I say clinical diagnosis in a clinic, you actually collect a sample, and then that sample is typically blood. Okay. Otherwise, in olden days, if you remember, people with diabetes used to give their urine sample. Urine is such an easy sample to collect and also dispose. You can simply dispose urine into sink, whereas blood, you cannot dispose blood into a sink. Okay, so that's a hazard actually. So otherwise, one has to really go for biopsy, as I said, endoscopy. Endoscopy is something used to actually see the internal organs, okay, and then also take a sample from the suspected region. So that biopsy, for example, if you want to find out if there is an infection or if there is any cancerous growth, you do a biopsy and find out whether it's cancerous or malignant or benign. Malignancy means cancer. And then if it is an infection, if you suspect that it's an infection, you actually subject it to certain culture techniques and then find out, depending on the bacterial growth, colony formation, and their shape, size, staining, other observations, you will come to know what kind of bacterium it is. Okay, so That's how it's possible. So, and then subsequently, you do have several other classes of uh, samples. Sputum is one sample. Sweat, of course, we still don't know how to use sweat so efficiently to diagnose disease, and saliva. You might have heard a news that now in US saliva is being used in place of this nasopharyngeal swab for COVID diagnosis. Okay, so saliva is such an easy sample. You can actually get a small sample of saliva into a small tube without any effort, and it is definitely not as painful as nasopharyngeal swabs. Okay, so these are different samples that you can access, but most of the times we talk about blood. So as far as constituents of blood is concerned, these are all the constituents of blood. And then what you have really is many times you have to quantify each and every of these things. Blood gases, for example, you have oxygen and carbon dioxide dissolved in blood. So these are the blood gases. So you want to quantify these two, partial pressure of oxygen, partial pressure of CO2, right? So why? Because elevated levels of oxygen is not good, elevated levels of CO2 is not good, right? In blood. And if you don't have enough oxygen dissolved in blood, tissue doesn't get oxygen and then tissue cannot survive. If you have less of carbon dioxide in blood, that also actually causes an imbalance in the blood pH. Okay, so one has to really tightly maintain the pH of the blood. So you have to have this dissolved gases also maintained very precisely. Electrolytes, potassium, sodium, chloride, these are some of the examples of electrolytes. Whereas metabolites, if you say, there are about 4,000 metabolites that are of interest to us. Okay, there are about 4,000 metabolites that are of interest to us, mainly because people have realized that it is not just genome, or it's not just protein or proteomics. It is the metabolites that also have a role in health of the tissue and health of the human being altogether. Okay, so that's the reason you hear now, since almost 10 years, you are hearing this word metabolomics. Okay, and of course, proteins are disease markers. Many times you can use certain proteins as disease markers. An example, if you have CRP, C-reactive protein, okay, that is a marker for inflammation. If a person has got an elevated level of CRP in blood, that means there is somewhere some inflammation in the body. Okay, so that's a very good indication. But where exactly, you don't come to know. Okay, for that reason, for example, if it is inflammation is in heart muscle, okay, then what would you do? Is a big question, right? So for that reason, people have started looking at certain particular biomarkers, certain particular proteins that are very specific to this particular disease condition. For example, if at all there is heart muscle damage, there is something called as troponin, cardiac troponin 1. Okay, so this is used as a marker. Elevated levels of troponin indicates a muscle damage and repair process that is in progress in the heart muscle. Okay, so that's most important. You have to realize that. So, like that. So I'll come to other things. So, for example, last but not least, uh, in fact, this is the first thing that people would like to do is complete blood count. Okay, so you're aware of this. What is complete blood count? And the cell constituents of the blood, RBCs, WBCs, platelets, right, and plasma. And then Blood count, why is it important? So, for example, if the RBCs are low in number, there is not enough oxygen supplied to the tissue. If the white blood cells are high in a particular sample, that means there is some kind of infection, right? So you typically know what is the condition. For example, platelets are important. If a person is actually having dengue, the platelet should not drop, count should not drop. So that becomes quite important. Fibrinogen is one of the big protein present in the blood. So as far as serum is concerned, of course, it has electrolytes, glucose, and then it has proteins, several of the proteins, and then it also has something like enzymes, and then many other molecules. So diagnosing any change in RBCs, WBCs can be related to a particular disease. Any change in any deviation in glucose levels can be pointed to diabetes. Any change in this bile acid, that can be related to liver. Right? Any change in the shape of the uh, white blood cells, so that is something like leukemia, right? So these are some things that you are very much aware of. You don't want to deal with these things. But you must understand one thing: what is the instrumentation behind this one? Because this topic is more about medical devices and then instrumentation. So, what is the existing practice to actually that gives you a complete blood count? In, let us say, you give the sample and then you get the result by evening, right? So, how does it work? So, typically, sample is collected at the collection center and then it goes to the central laboratory. In the central laboratory, all the samples are just put in place, and then what they do is typically they use automated means of quantifying the blood sample. Okay, and then each and every particle number in a given volume is quantified. If you want to do that by manual means, it takes a lot of time. That's what people used to do about at least 10 years ago. Now nobody really does it unless it's very important. So yeah, important. So you have carbon dioxide and oxygen. How do you basically get these partial pressures of these gases quantified? So before you can actually talk about partial pressures, there is something always most important: arterial oxygen saturation. That actually points to the oxygen content in the blood. Okay, so nowadays with COVID, this has become quite important. It is somehow observed that most of the COVID infected people really have a little low level of oxygen saturation. Okay, so not all. We really don't have the data. It may not be all, but it may be at least some people. Right? Okay. I'm just trying to do this. I'm not able to. So, as I mentioned, the pH is typically maintained in a very narrow range. This is the partial pressure of CO2 and O2. I'll leave it to you. I can share the PPT later. How do you quantify this blood gases? It's quite simple. You just have to use a galvanic cell. So on one side, you use a standard electrode. On the other side, you use an electrode where the electrode is coated with HCl and then placed in HCl, and then it is open to interact with the environment. So this way, you have pH measured very easily in terms of voltage readings. Potential difference between these two electrodes gives you a voltage reading. And this voltage is proportional to the pH. Now, how do you quantify O2 and CO2? Okay, so when you do that, you can actually, especially when you have CO2, you will be able to use the same electrode, just like a pH electrode, and you can be done with it. So the same electrode can be used for quantifying CO2 as well. Only the difference is you typically have a semi-permeable membrane that can allow O2. Sorry, CO2. So this is for CO2 sensor. But this is more like potentiometric sensing, whereas for O2, you really can't have such a nice equation. So that's the reason you see involvement of an external power supply. Whereas here you don't have any external power supply. Whereas for detecting O2, you need this external power, and then this electrode actually operates as a amperometric sensor. So this is how these electrodes look like. Okay, CO2 electrode looks like this, almost like similar to pH, whereas oxygen electrode somewhat slightly different. So this is a PPG machine or pulse oximeter that is now widely being used. Right here, what you see is a wired device. You actually have very well wire-free device also in the market. So it works on 2 LEDs and 1 single PD. So 2 LEDs are chosen such that the difference is at one wavelength between oxy and deoxyhemoglobins is not much, but whereas at other wavelength is very high. Okay. Coming to electrolytes, Nernst equation, electrolytes are very helpful in maintaining the body fluids balance, and then blood, right? And also, in order to carry out electrical signals, for example, you need to have somewhere you have brain, and then my hand, the signal has to reach my hand, right? So how does it happen? It's mainly because of the charges. So this is also more or less similar to similar electrochemical technique. Here you have, however, something called as ion selective electrodes. Okay, so, most important. You have to select the ion of interest, whether you want to quantify potassium or chloride or sodium. What is it that you want to quantify? You should be able to have a membrane, a membrane that makes the electrode selective to only to those particular ions of interest. So you follow the Nernst equation, and you get this particular equation where this activity is proportional to the potential difference. Ion activity. So metabolites. What is metabolite? The entire food spectrum of food that we take inside is metabolites, and then air we breathe is also metabolite, right? And then pollutants that we take are metabolites. The drugs, the chemical drugs that we take when we are not ill, they are also metabolites. All that, right? So in general, you will be able to classify uh, the blood, the metabolites into these 4 categories: sugars, nucleotides, amino acids, and lipids. Okay. So what is typically metabolites? It in fact has a molecular weight less than 2 kilodaltons. And more important, here you notice that there are about 4,000 metabolites that are of interest. So how do you separate them? In case of electrolytes, in case of gases, you have used ion selective electrode. But you cannot really apply the same technology to metabolomics. Why? Because that won't work. So how do you do? Is the standard procedure is nothing but use GCMS. GC stands for gas chromatography, and MS stands for mass spectroscopy. Excuse me. So here, when you actually, in fact, in the principle of chromatography is shown here. People who are not aware of chromatography, they can pay attention. When you actually drop a sample in and pass it through a medium, a polymeric porous medium, so whatever that biochemical that you have introduced, actually it can it will move forward based on its molecular weight and charges. Okay, charge with respect to the matrix material. So after that, you collect it into this sample container and then push it through whatever mass spectrometer. So these are typical spectrum of metabolites that one can see. If a person takes the metabolomic graph, this is how it looks like. So whatever is numbered that you see is actually a compound. Yeah. Okay. So any questions so far? This is almost halfway through. Any questions so far? These are more fundamentals I thought I will cover. Any questions so far? No. Okay. Now, how many of you are aware of ELISA?
Myself, sir.
Yes, sir.
Yes.
Yes, sir.
Yes. Very good. So, how many of you are aware of chemiluminescence immunoassay? Chemiluminescence immunoassay.
I have seen my PhD scholar work on that, sir.
Okay. Great. Where?
At DRDO in Mass.
Okay. Sure. So, ELISA, you are very much aware of it. This is still in textbooks and it is still used in research labs. So that's the reason we're still reading it, but that's more fundamental. CLIA is a little advanced version of the same procedure, but a few chemicals have changed. The concept is actually different. So the concept here is chemiluminescence immunoassay. Here you don't really have any enzyme, but you actually have luminescence. So in ELISA, you actually read color, a drop in light intensity due to color formation. Okay. Whereas in CLIA, chemiluminescence immunoassay, you see instead light generated as a result of the biochemical reaction. Okay. So that's how CLIA is most powerful, and then being currently used in all diagnostic labs as far as protein diagnosis is concerned. Okay. So most of the times when you talk about proteins from clinical diagnostic point of view, you have to see that how well you identify a disease marker. Okay. So the first and foremost question, if you are actually looking at trying to detect a disease, identify when that sorry, I lost my flow. So when you want to identify a disease, identify a particular protein that is relevant to this disease. In case of a disease condition, if this particular protein goes to elevated levels, that means this is your disease marker. Same should happen in almost several other subjects also. Uh, and that's how you, if you can identify a protein, you actually will be able to develop an immunoassay for that. Okay, so that is how a disease markers are basically identified. Identify disease patient, perform. You take the collect the sample, you perform LCMS, and then identify the protein biomarkers, generate antibodies, and then generate diagnostic immunoassay. So this is the procedure that you typically have. But what you see in labs is this: perform diagnostic immunoassay. That's it. But you really are never ever exposed to these four parts most of the times. Okay, so this is more fundamental in nature. At least the first three are really fundamental. Fourth one is more like industry job. It's not something do with the intellectual things. Okay, so ELISA, you are very much aware. So there is a sandwich immunocomplex with the secondary antibody having labels. In luminescence, you have instead of an enzyme in electrochemiluminescence ECLIA or CLIA, both are more or less similar. That's actually still an enzyme, but the substrates are going to be different. Okay, so that's what makes it different. So coming to biosensors, I'm sure you have got introduction from many people. I don't want to cover. And DNA markers, of course. Okay, here is something. How many of you are aware of lateral flow assays? How many of you are aware
I
of?
am, sir.
Sorry. Yes.
I have worked with lateral flow assays.
Okay. Do you know how they work? How many of you don't know how a lateral flow assay works, LFA works? That means all of you know about it, right? Yeah. Okay. Now, what is this color because of? Can you tell me? Am I connected? Hello?
Students are interacting with you only. Please reply.
Sorry?
Students, please connect with the sir. Sir is asking to you only.
Okay, fine. So I just have only one question. Why this color? What is this color? What is the material? If you want, probably you can use the chat box, maybe, if you want.
So these colors show because that chemical, particular chemical is reacting with that.
Okay. Any other guesses? Do you have any nanotechnology here?
Yes, sir. That's why nanoparticles sensed by the sensor.
Okay. So this. Okay. Right. Well, these are actually because of gold nanoparticles. You see this color. For example,
Yes, sir.
if you put a dye, a normal dye. Okay. Do you see such a color?
Sir, not exact that color because every dye have their own color.
Right. Absolutely. But there is something called as extinction coefficient or absorption coefficient, more precisely. So, can you compare the absorption coefficient of small molecules such as methylene blue and then compare it with gold nanoparticles?
Sir, gold nanoparticle is working better than the ethylene blue.
Yeah, that's true. But how better, you must know. It is 10 power 5 times 10 power 5 times
Okay.
better.
Okay, sir.
That's the reason pregnancy test kits also use gold nanoparticles. Okay. Nowadays, there is some more research going on to replace these gold nanoparticles with fluorescent beads. That is going on, and people are working on it. Hopefully, we'll see, but
Okay, sir.
not in the home care, but in the hospitals. Okay. So, well, you have DNA. How many of you? I'm sure you all are aware of PCR, polymerase chain reaction for DNA amplification, right?
Yes, sir.
But do you know? Do you know what is molecular beacon? How many of you are aware of molecular beacon? Okay. So molecular beacon is what you see on the left side. Okay. So here you have two things. One is a quencher and a reporter on either end of the DNA strand. This is completely one single stranded DNA. Okay. And this single stranded DNA has sequence such that this region is complementary to this region, and that's how you actually have a small hybridization found. Okay, so the two ends hybridize, and then you actually have this quencher and reporter coming together. But however, you have the loop region. This is the loop region sequence, single stranded DNA sequence that is probably complementary to something of interest to you. Okay, a target analyte, a target DNA of interest to you. What happens is this portion of this loop sequence typically forms gets hybridized, and then it actually breaks the stem region. So what happens is simply the force in this region is so strong that these hydrogen bondings between ATGC pairs typically cease to act. Okay, they're pushed apart. And then the reporter and the quencher. So the reporter is very much away from the quencher. You start seeing the fluorescence the moment you shine light. Whereas here, in the moment if you shine light, you still don't see anything because reporter molecule is in close proximity to the quencher. So this is being used in Gene Expert. Uh, it is Gene Expert is a machine for diagnostic for tuberculosis diagnosis. Okay. With that, I stop. Of course, toxins and foreign substances, you are very much aware of it. But one of the most important problems that we have is when it comes to okay, when it comes to blood gases or electrolytes or metabolites, the concentrations of the analyte that you have are at least in micromolar regions, micromolar range. Okay, did you get my point? When you look at these concentrations, blood gases, electrolytes, metabolites, they are very nice at very high concentrations. Whereas many times the proteins, which are disease markers, they are at low concentrations, sometimes picogram per ml, femtogram per ml. Femto means 10 power minus 15 grams per ml, or minus 10, minus 9, or 1 nanogram per ml kind of concentrations. So many times these conventional techniques may not really work. So for that, you need higher uh, high-end technologies. Okay, so that's where biosensors come into picture. Right. So with that, I would like to stop. So let me stop this. Do you have any questions so far?
No, sir.
No. Okay.
No, sir.
Okay. Great. So I'll have to share screen again. Post disable.
Sir, in the meanwhile, I have a question.
Yes.
Apart from proteins, when we speak about metabolites like vitamin B9 or vitamin B12, we have a blood amount of nanograms per ml.
Sometimes these vitamins are in nanogram.
So
Yes.
it is easier to measure some other metabolite like
Just a
glucose,
minute.
which is in a larger concentration, but when it comes,
Sorry, your voice was breaking. I'm sorry. Could you please repeat the question?
uh, yes, sir. Am I audible now?
Yes.
Yes, sir. When we look at biosensors, especially while estimating the metabolites, glucose and all has a larger concentration than a lot of other vitamins like B9 and B12. So
That,
these B9 and B12 are in nanogram per ml concentration. But
right.
when we speak about microfluidics and biosensors, is
Right.
it possible to measure it accurately with a very small amount of current that is generated? For example, when I use an amperometric sensor.
Right. Okay. Here is the thing. It is possible. Let me tell you that the possibility is there. So that's what is called as proof of concept. In fact, a proof of concept has been established by several people already for analytes that are much smaller in concentration. Okay, and not may not be just vitamin B12 or these kind of things. Vitamin D, or example, for example, there are many other analytes that are actually many times less than 1 nanogram per ml, and people have established a proof of concept using amperometry or optical techniques that it is possible to detect such concentrations in a laboratory. The problem comes here is uh, when the technology that is demonstrated at the lab level, when it goes to scale up, okay, many times these technologies that are being developed in the lab probably have several limitations, and the reliability is also probably an issue. That's the reason they all fall apart. So far, you really don't have very good alternative technologies in comparison to CLIA, and then a simple, colony counting or whatever. You really don't have very good technologies in place. So that's what I would like to take. Everything is possible at the lab, but scaling, reliability, these issues, once they are taken care, then the product is in the market. Okay. So yes,
Yes, sir. Thank you, sir.
thank you. So when I tried to share, I actually seem to have a bandwidth problem. When I was trying to share, I was not able to hear properly. I hope now I will be able to do that. So where is the PPT? Okay, here is the PPT. Share. Okay. Are you able to see the screen now?
Yes, sir.
Great.
Yes, sir.
Good. Now, so here I'm trying to give you an example. Okay. Don't take this as uh, already existing or anything. It's an example that we are. Uh, it's an example that shows how to develop a biosensor. Okay, because it's a school, whatever webinar or school model that you have, and you need more fundamentals than advanced topics. I will basically run through how a biosensor development can be taken up with a given idea that you have. Okay. Fine. So we have been working on fiberoptic biosensors for a long time. At least in IIT Madras, we have completed 9 years here, and in this 9 years, we have come up with uh, one or two different technologies that really are showing some promise. So we have got a DST project, a CRB funded project, as well as a Indo-US endowment fund project to develop COVID diagnostics using this fiber optic biosensor platform. Okay. So having said that, I'll just tell you, I'll I'll show you how some things can be realized. Of course, this our research lab as such, you know, the focus is, okay, what is this? Okay. The focus is more on bringing the laboratory technologies to point of care. That's what the focus is. Or if not point of care, at least can you minimize the cost so that they can be installed in our primary healthcare centers. Currently, the clinical diagnostics market is completely driven by uh, economics of scale. Okay, do you understand what is meant by economics of scale? For example, if anybody gives a blood sample, all the way this blood sample goes to a central laboratory, and there you have many samples pulled up, and then you actually have hundreds of samples analyzed together, because you actually have uh, your tests become cheaper when you have large number of samples to be tested. So this obviously use a lot of expensive equipment, but the running cost is really low. So, but establishment cost is pretty high. But what it requires is transportation of sample. So imagine you in Chennai or Delhi, and imagine you in somewhere Koraput. You know where is Koraput? If you
No, sir.
no. Okay. There is something called as Koraput and Kirandul. Those are agency areas. They were not even touched by British. They are in the state of Orissa, but the access is actually through Vizag. So you, in fact, you have a good uh, track built from Vizag seaport to all the way to this Koraput and Kirandul to take iron ore out for Japan. Okay, so these are quite remote places.
Okay, sir.
This is just an example I'm just citing. You have several such remote locations still not really accessible. Now, if at all you want to conduct some simple tests for a person sitting over there, for example, a person there says, okay, probably he has some radiating pain in the left finger, left ring finger, or left small finger. Probably you suspect that it's a symptom of myocardial infarction, but you really don't have a means to test it, right? Mainly because you don't really have something that you see as a home-based device or anything like the point of care device, so sensitive, right? Still, there is a problem. If it is glucose, well and good. Pregnancy test kit is also still okay, available, but many diseases cannot be diagnosed using the existing technologies such as these. Okay, so there is always a need for new technology, and that's what this PPT tells you. One example of them. So we use optical fiber to develop sensors. Optical fiber and noble metal nanoparticles, mainly gold nanoparticles. They are also called as plasmonic particles. Okay. So what happens is, if you can actually develop certain sensors, one technology, but can be useful for many applications. Not only clinical diagnosis, but environmental monitoring, food industry. Food industry, for example, bacterial contamination of food, or fungal contamination of food, or in pharmacy, agriculture, right? So that's all possible. So here you are aware of this lateral flow assay. It's nothing but a sandwich immunoassay with gold nanoparticles. But you also need to realize that many times quantification is a little difficult. Okay, sensitivity is poor. For example, nowadays the COVID diagnosis is actually COVID antigen diagnosis is nothing but based on these lateral flow assays. One of the major issues is they are not really so sensitive. The manufacturer claims they are super sensitive, their sensitivity is more than 90%, but the ground reality is different. This is mainly because this technology is very simple, but anything so simple has its own limitations. So one thing, if you take an optical fiber, how you can actually make use of an optical fiber for developing a sensor. Why I'm talking about this is, this is a gold nanoparticle. So the idea is, will you be able to use this gold nanoparticle in the same assay, but detection of this gold nanoparticles more efficiently? These optical properties of this gold nanoparticles, they absorb light at green wavelengths, for example, 520 nanometers. Okay, so if you shine green light, the green light is actually absorbed. Whereas if you shine red light, the red light is typically reflected. Okay, so that's the property of these gold nanoparticles. So use an optical sensor to actually quantify the gold nanoparticles more efficiently. So one can actually make use of optical fibers because these optical fibers are very cheap, and then one can actually miniaturize the sensors. If you can recall some physics that you have done in your +2, there is something called as total internal reflection, right? So what happens is there's always some amount of light that is leaking at the surface. So this is an optical fiber with a core. So what you do is you declad, that is, you remove the clad layer on the core, put your antibodies, and then you start seeing when antibodies come and bind, you can actually see a change in the light propagation, light property. So I will not bore you with these details, but I'll tell you one thing. This is how a U-bend optical fiber can look like. This is 200 micron core diameter. Okay, that's called as multi-mode fiber. In fact, these fibers are different from the ones that you typically have in home internet networks. They are single mode fibers. Okay, whereas what you see here is a multi-mode fiber. That means they are, for your own understanding as of now, they carry more light, multi-mode, more light in different modes, and then the diameter of the fiber is much higher. So that's what you have to understand. 200 microns, whereas single mode fibers are only 8 microns. So what happens is, once you functionalize these probes and dip it in a gold nanoparticle solution, these gold nanoparticles find affinity to go the amines on the surface, and they start sticking to the surface. And over time, let us say here 5 minutes, 10 minutes, 15 minutes, like that, up to 1 hour, if you observe, there is a huge light that gets absorbed. So you pass light through this LED on one end and detect the light on the other end using a photodetector. So if you use a spectrometer on here, okay, uh, so you will be able to measure the spectra, and then you see that these gold nanoparticles can be detected very efficiently using these U-bend probes. So we have used that technique and ran some assays. So this is a simple setup. You have an LED, you have a photodetector, and this is a U-bend fiber. Okay, this is a blue thing is buffer, and the glass is here at the bottom. So you actually, just like sandwich immunoassay on a paper, you actually run a sandwich immunoassay on the U-bend fiber surface. Okay, so what happens is you typically get a very good response, very nice response. In fact, this is with human IgG concentrations. We were able to go down to 1 femtogram per ml concentrations in 25 microliters. That means about 100, 150 molecules in the 25 microliters, which is not a joke, right? So it's a combination of a good sensor and a good nanomaterial that gives you such a nice result. And the instrumentation is not very complex here. It's a simple off-the-shelf LED and off-the-shelf photodetector, nothing else. Okay, we have done some more characterization and proved that this is indeed the response. And then one can actually change the dynamic range because one of the important aspects with clinical analysis is sometimes your analytes are in a nanogram per ml concentration, sometimes in picogram, and sometimes they're in femtogram per ml concentrations. So you should be able to tune your dynamic range of the sensor. So you will be able to do that using by choosing different particle size. You'll be able to do. So that's the advantage of this. And we have compared our results with there is something published in Science, of course, about 15 years ago. When people have to, without a sensor, you have to do 6 steps to get the same result. We are able to do it in 2 steps, mainly because we are using an efficient sensor. So you realize, so I would like you to appreciate that you, on one hand, people know how to use nanomaterials, but if you can actually use certain transduction mechanisms, both of them put together, they give you wonderful results. Okay. So some SPR-based characterization, not really necessary. If I use some literature report, not necessary. Okay. WHO, if you are aware, WHO has got a criteria called as assured criteria. WHO here, World Health Organization, and they have issued a criteria called as assured criteria for HIV diagnosis, especially for African countries or low-income countries. So what is this criteria? So A stands for affordable. That means it has to be affordable to the population of interest. Sensitivity should be high. Specificity should be good. User friendly. So it's like, well, users, you cannot expect users to do a lot of processes before they can get a result. So that is user friendly. So minimal steps. And then rapid. So typical time that you may actually have is 10 to 25 minutes of time, detection time. And more importantly, equipment free. If you need a big equipment, then it's not really going to work out. Mainly because, you know, the cost adds to it. And if you have an equipment, it needs to be powered with power consumption is going to be high, and then you have to provide enough power, enough power backup. All these are issues. So if you can actually have a handheld device more like a torch, and that's nice, right? So we have actually went ahead and tested this. Now, apart from HIV, another uh, important disease that is plaguing the poor man is tuberculosis, Mycobacterium tuberculosis. So we have applied the same assay for tuberculosis diagnosis using something called as MANLAM, mannose lipoarabino mannan, okay, in urine. Why urine? Urine is so simple to use. You don't need sputum. Many times, if you take some patients, even infants, or extrapulmonary TB, that means TB not in lungs but elsewhere in the body, maybe bone, maybe in colon, maybe elsewhere, maybe brain. So they don't really develop so much of sputum as pulmonary TB patients do, right? So sputum is really ruled out, whereas urine is a sample, very neat sample. It's a safe sample. Disposal is easy, unlike a sputum sample, right? So many advantages. So, but only problem is this lamb is at concentrations, a wide range of concentrations, starting from 1 femtogram to all the way to 1 picogram. So it varies depending on it varies between person to person. Okay, so one has to devise this. So what we can do is we have actually shown that this man lamp can be detected down to 1 femtogram per ml. So that's one. We are happy with this development, and we actually want to go ahead in this direction so that we will be able to make devices and use it for public good. So, of course, there are many other things going on in the lab. In the interest of time, if you have more questions, if you have questions now, we will take up those questions. Otherwise, we'll go ahead. I have time, another 15 minutes time. So any questions that you have so far?
Good afternoon, sir. This is Yogesh again.
Yeah.
Regarding the light source in this fiber optic sensor, how
Right.
reliable is a light emitting diode? Because the light emitting diode has its own signal to noise ratio limitations throughout its lifetime. It varies with its usage. So, how
Right.
reliable can we use that?
Very good question. So when we talk about a light source, yes, you are right. When you switch on the lights, for example, our home LED lighting systems, the variation, you do see a lot of variation over time. But the ones that are being used for scientific applications, you really don't need to operate at such high powers, in fact. So what happens is when an electronic device is operated in, let us say, 10 milliamperes, especially LEDs, if you can operate the LED at 10 milliamperes or less than 20 milliamperes, the heat dissipated is very low and the fluctuations are going to be low. And also on top of it, you typically have certain electronic circuits such as constant current circuit, and then on the photodetector side, good amplifiers, simple amplifiers, okay, not expensive. With that, you will be able to maintain the light intensity constant over at least 1 hour. So we have seen this and we have observed, we have done all these experiments using this LED only. For example, what you see is absorbance at, it's not 520, it's 525, not 542 nanometers. It's a small mistake here. Okay, we see that absorbance, and you see that when there is a control sample, this black sample, you don't really see much difference, right? Whereas when you have, let us say, 5 femtogram per ml or 10 femtogram per ml, you see a clear difference. And here also, you don't really see any fluctuations. And no data processing is being used here. This is all raw data. What I'm showing is raw data. So what I want to convince you is these nowadays the LEDs and photodetectors, the optoelectronics has actually taken a good advancement, and you do have stable light sources possible. Okay, we are using LEDs. In fact, LEDs are much better than any other light source. For example, halogen lamp or incandescent lamp, or for example, forget about these CFLs, right? CFL are like they fluctuate a lot, but LEDs are very nice. You will be able to use, you can use these for scientific applications, technological applications. Yeah,
Yes, sir. So, as a follow-up question, as
right,
an alternative to LEDs, currently some groups are also using commonly available lasers like pointers. How efficient would that be in the same place, sir?
right. A very good question again. Um, One of the major issues with lasers is the laser power actually is not as stable as LED. So when an LED is sufficient for your needs, don't go for a laser. Okay. So now LEDs are actually having a good enough optical power. You do get LEDs with 1 watt typical, or 2 watts, like that. So that is sufficient for these applications. Of course, you just have to have a good heat sink also sometimes, right? But as long as you operate the LED in less than 10 milliamperes, there are no fluctuations as we see.
Okay, sir. Thank you, sir.
Thanks. So, any other questions on tuberculosis diagnosis? Did you understand the concept why one has to go for an antigen-based test even for tuberculosis, not only for COVID nowadays? Of course, I'm referring to COVID all the time because you will be able to relate it. So that's the reason. Okay. So I still have some time. I'll go ahead. Fine. Okay, I have another question for you. Why do you use water purifiers, and what do the water purifiers do?
That water purifiers filter the unwanted metals from the water, which are harmful for the human body.
Right. Okay. So they do have some filters that can actually filter out some heavy metal ions. I'm not sure to an extent, but it's a reverse osmosis process that actually takes care of most of the things. It's not that mainly they have any
Yes, sir.
filters. It's RO, RO filter you
Yes,
call
sir.
it as. That is
Reverse osmosis.
right. So, what about UV water purifiers? Now you have UV water purifiers, you have UV RO. What is UV doing there?
UV is
So
essentially
they,
to destroy any kind of pathogen or bacteria in the water.
right,
Right,
right, sir. So they are
right.
killing the that
Yeah,
microorganism present in the water.
right, exactly. And what is the light that they use? They use typically 250,
UV light.
yeah, UV 254. You have a UV lamp emitting at 254. It's okay, it's affordable also. It's not a big deal. So they can use that. And then 250 nanometers, 254 exactly nanometers, all these pathogens do absorb uh, light. You see that? Okay. They do absorb light in a significant way. So will you be able to use this property for sensing the pathogens? So look at the phenomena, and if you want to develop a sensor, you have to have a good understanding of the phenomena. So from science, you have sensors. In order for you to develop a good sensor, you need to have a good understanding of the science behind whatever we do. Okay. So here is the science. Is what you see on the left bottom corner is a spectrophotometer data for E. coli and then human IgG. So you know that proteins absorb at 280 nanometers. Of course, pathogens do absorb beyond less than 300 nanometers, right? So will you be able to use an optical fiber uh, using this property, UV LED property at 280 nanometer, and detect pathogens? Well, it is possible. We have shown that long back in 2011. So once the pathogens come and bind, the UV light, just like the nanoparticle, absorbs light when it's sitting on the UV region. When a pathogen comes and sits on the fiber surface, the light passing through this waveguide, the glass tube, typically is absorbed by the pathogens. Okay, so that's possible. So that way you will be able to do. And here is another example. So bacterial toxins, LPS, lipopolysaccharides. You know, uh, you have this LPS-free water. For an injection, if you want to give an injection, you typically have this toxin-free water or LPS-free water. Why? Because this LPS, if there is any LPS in water, it can actually cause disease. Okay, toxin, right? So, how do you detect this LPS? This is something is again another example. You have a receptor based on hydrophobic interactions, and then your LPS looks like this, and then you have PMB polymyxin B. It's a antibiotic. Okay, coated on top of nanoparticle, and you get some kind of PMB basically goes and binds to the core region of LPS. So you actually kind of have a sandwich assay by which you will be able to detect LPS if present. So We did that on an optical fiber. The optical fiber has to have some long chain molecules, a hydrophobic long chain molecule. Okay, you can call it as alkyl silane, C18 chain. So this is a carbon, each corner is a carbon. Okay, you have 18 carbon chain here. So we did see that one can actually detect LPS down to almost 0.1 nanogram per ml using this approach. So these are gold nanoparticles that enable this. Okay. So we are also into technology development. We want to develop technology. So we started bending the probes. We made machine for bending the probes. This is a third generation bending machine developed in the lab. So these are rotating mechanism. These are laser, CO2 laser, what you see here. So CO2 laser falls on it, and this is the process that happens here. Okay. So this is an automated U-bend fiber-proof fabrication method technology that is developed in-house. We have been developing our own light sources. So we are putting LED and photodetectors together to realize a point of care diagnosis. This is a very crude assembly way back 4 years ago. And then we also made some refractive index sensors. We applied it for milk quality check. So if at all you add water to milk, too much of water in the milk, this fat content goes down. So if the fat content goes down, the sensor is able to pick up the response. Okay. So the idea is now what is under development is we are developing an array biosensor. So you see that these are 8 probes, okay, 8 U-bend probes. And these 8 U-bend probes are here. And then on one side you have a light source, and on the other side you have a photodetector array. So everything put together. So this is, he is our senior project officer, an expert in robotics, programming, and mechanisms. So he has made this, I would say, equipment, small hand benchtop equipment. So we are going ahead for this testing now. So this, what we can do is different possibilities. Label-free sensing possible. Label plasmonic assays, for example, LFA. Whatever happens in LFA, you can do that. You can actually have bacteria detection without any nanoparticles. All these are possibilities. So we are exploring those. So what is ahead? We want to go ahead with tuberculosis testing and myocardial infarction as we go along, and then pathogen testing, everything, device manufacturing. These are in progress. So with that, I stop. I thank my group. This is almost time, 12:20. I can take questions for 5 more minutes, and then we can wind up. Any questions? So remember one thing: if you are developing technologies, there's no one who would like to take it from you. That's what we are saying. We have to take it to a level where a company can absorb it. Okay, so that means you have to have a startup. You have to basically run around, have things together. Of course, it also requires a good amount of funding, right? And then it also requires a lot of perseverance. Funding is one thing, and then having right people is another thing. So everything has to come together to realize a good technology. Okay. So